Hello, everyone. Thank you for joining our webinar today. Before we begin, I have a couple of announcements. First, the audience will be muted for the duration of the webinar. If you have a question, please use the chat or Q&A function to the right of your WebEx window, where my colleagues will be able to address your questions in real time. In the event that we have not addressed your question, we will follow up with you offline after the webinar. Second, the webinar is being recorded, so in the event you can't stay for the entire duration, we will have the recording up on our website. And with that, I'd like to begin on some tips, tricks, and troubleshooting the RNAscope and BaseScope assays. Here's a quick overview of the topics I will be covering today. I'll do an overview of the RNAscope technology, performing the RNAscope assays, understanding the RNAscope assays, and then we'll delve into some troubleshooting. I'll also address doing the dual ISH and IHC workflow, as well as a brief introduction to the Ventana universal assays. And afterwards, I'll address some frequently asked questions. To begin with the RNAscope technology, the RNAscope assay is an in situ hybridization method that utilizes a unique double-Z target probe design, followed by a signal amplification system, which will allow you to visualize a single mRNA transcript as a punctate dot through either chromogenic or fluorescent detection. This highly specialized system allows for simultaneous signal amplification and background suppression. This will allow you to directly visualize your mRNA markers in cells and tissues, and in a variety of different types of tissues, including FFPE, fresh frozen, fixed frozen tissues, as well as PBMCs and cultured cells. Here is a brief overview of the RNAscope method. It is a slide-based assay intended for use on the same types of samples you would use on either IHC or immunofluorescence, including FFPE tissues, fresh or fixed frozen tissue sections or cells. The RNAscope methodology involves pre-treating the tissues or cells in order to unmark the target mRNA. With the target-specific double-Z probes, you will then hybridize to your target RNA using the oligonucleotide probe. The target probe is a pool of oligos that we've designed using established informatic tools to provide an assay which we will guarantee will be single target specific. Detection is then enabled through an amplification procedure that involves sequential hybridization steps to build up a tree-like structure shown here schematically, which I will describe in more detail on the following slides. Through this method, we are able to localize a tremendous amount of signal to an individual transcript to the point where we can actually detect and visualize individual transcripts as dots in a field of cells. This visualization of punctate dots allows for quantification of gene expression detected by the RNAscope or BaseScope assays. The first key feature of the RNAscope technology is the probe design. We depict the oligonucleotide target-specific probes as Zs to emphasize the fact that they have two regions linked by a spacer. Each one of these oligonucleotide sequences has been designed using an informatics algorithm that selects sequences to specifically bind physically to the target sequence and not cross-hybridize with any other sequence. The bottom of the Z complements and hybridizes to the target transcript. For amplification to occur, two Zs must hybridize to the target sequence right next to each other. Once this happens, this creates about a 50 base target-specific binding site on the bottom of the double Z pair. The top of the Z is the base for the amplification structure. When two Zs hybridize, it creates a binding site upon which a preamplifier can bind and the amplification tree can be built. A standard RNAscope probe for a target sequence of 1,000 bases or more will consist of 20 double Z pairs pooled together that are designed to hybridize next to each other along a target region. This allows for a tremendous amount of amplification and signal potential. However, only a few ZZ pairs are needed to bind to the target RNA sequence in order to generate enough signal for molecular detection. Here's a schematic showing the hybridization and subsequent amplification of the RNAscope assay. The double Z probe will bind to the target mRNA transcript. Once the double Z probe binds, it serves as a foundation and binding site for the preamplifier molecule. Once the preamplifier is bound, it serves as a binding site for the amplifier molecules, and each amplifier can further bind multiple labeled probes, sequentially hybridizing to assemble a branching complex at each double Z binding site. Labeled probes can contain a chromogenic enzyme such as horseradish peroxidase or an alkaline phosphatase that generates a visible signal after chromogenic reaction that can be detectable under a standard bright field microscope. The labeled probes can also contain fluorophores that allow for visualization of the signal under a fluorescent microscope. The signal amplification strategy allows for visualization of target mRNAs as a single dot, where each dot represents an individual RNA molecule. Specificity is shown here if the single Z binds to the target mRNA transcript that the preamplifier cannot bind, as it needs both Zs to provide a binding site. This helps to ensure the specificity of the probes and to reduce potential background staining. To date, we currently have over 1,200 peer-reviewed publications using the RNAscope and BaseScope technologies. This has shown an increase in the market adoption of the RNAscope and BaseScope assays utilized in many different research and application areas. This slide presents all the assays that are currently available from ACD. If you have a single ISH target, you may choose our singleplex assays, such as the chromogenic brown or red. If you have two targets, you may choose the chromogenic duplex assay, seen here as a red-green combination. However, if you have 3 or more targets, you may opt for the multiplex fluorescent assay. If the target of interest is a shorter sequence, if you're looking at a splice variant or exon junction, then you would consider the BaseScope assay. Do note that some of these assays are available as well through an automation platform using the Leica Bond RX or the Ventana Discovery platforms. This is a diagram to show the singleplex assays, which includes the amplification tree, which is then tethered by an enzyme. In this case, the red assay utilizes alkaline phosphatase, which can be visualized by a fast red chromogen. Likewise for the brown assay, which utilizes a horseradish peroxidase and visualized by the DAB chromogen. A brief overview of the duplex amplification, in which both channels have independent amplification trees. The C2 channel is amplified and utilizes the alkaline phosphatase enzyme and fast red substrate to visualize one mRNA marker. And a second amplification tree is built for the C1 channel, which utilizes a horseradish peroxidase enzyme and a green substrate to visualize another marker. You can note that the 2.5 HD duplex is automated. You can do this on the Leica Bond RX with either a red-brown or a red-green chromogen, or you can do this on the Ventana Discovery Ultra platforms as well using a red-brown combination. For the duplex assay, by default, our C1 probes are provided in a ready-to-use 1x concentration. The C2 probes then are provided as a 50x concentration, and so to make a duplex probe mixture, you would mix your C2 probes in a 1:50 dilution with your C1 probe. In the event you only wanted to look at your C2 probe, you could use a Planck probe or a probe diluent and then continue to make a 1:50 dilution. Here's the multiplex fluorescent assay, which again utilizes the same amplification schematic and similar to the duplex chromogenic assay, where it will build 3 independent amplification trees so that a C1 probe, C2 probe, and C3 probe each have their own amplification structure. Particularly, this is for our fluorescent multiplex assay, which has our label probes directly conjugated to fluorophores using the Alexa 488, ATO 550, and ATO 647 dyes. Similar to that of the Duplex as well is that the C1 probes are given in a 1x concentration and ready to use, while the C2 and C3 probes are 50x. So then to make your 3-plex probe mixture, you would use your C2 and C3 probes and do a 1:50 dilution in your C1 probe. And similar that you could use a probe diluent in the event that you only wanted to look at your C2 or C3 probes independent of your C1 probe. For performing the RNAscope manual assays, we'll go over a couple of tips. With the RNAscope assays, you may perform this as a 1-day assay, though there are options to split this into 2 days. The first option to stop the assay would be to complete the sample preparation and pretreatment, and then to begin with probe hybridization and detection on the following day. Please do refer to your appropriate technical note per your sample type to determine where the stopping point may be feasible. If you are unsure of this while reviewing your technical note or use your manuals, please feel free to contact technical support. The second option for splitting into a 2-day assay would be to complete sample preparation, pretreatment, and probe hybridization on the first day, and then incubate your slides overnight in a 5x FFPE or a saline sodium citrate solution. The following day, you would remove your slides from the 5x FFPE and wash with the RNAscope wash buffer, then follow with the amplification reagents. This is highly recommended to do with the Duplex and Multiplex fluorescent V2 assays, as these are much longer assays with many more amplification steps. The assays are completed in 4 major steps, which include pretreatment, probe hybridization, amplification, and detection. This particular workflow is for FFPE and fixed frozen samples, which require both a heat-mediated epitope retrieval and a protease digestion for pretreatment. Here's the workflow when using fresh frozen samples, which omits the heat-mediated target retrieval step. Please note that the assay flow is also similar for BaseScope assays, which we do not have a technical note as of yet for fresh frozen tissue, but note that it will follow the same workflow. If you have any further questions, again, please feel free to contact technical support. Here's a more detailed workflow for the multiplex fluorescent V2 assay. Similar to the previous workflow diagrams, pretreatment is completed first, followed by the target probe hybridization and subsequent amplification steps. This differs from the previous multiplex fluorescent assay, as each channel is developed sequentially. The AMP1, 2, and 3 steps are a pooled combination of the preamplifier and amplifier molecules so that the amplification trees for the different channels can be into form simultaneously. This schematic also shows that fluorescein, Cy3, and Cy5 are in the C1, C2, and C3 order respectively, but note that these may be swapped out per your preference. And please note for the multiplex fluorescent V2 assay, this is a TSA-based assay and requires the purchase of the TSA Plus or Opal dyes from Perkin-Elmer. In the event you have any further questions regarding the V2 assay, all of the pieces that you need, please feel free to contact technical support. This is a standard RNAscope kit configuration, which will include the necessary pretreatment reagents, the detection kit of choice, and the wash buffer. But not pictured here are the target probes. Do note with your wash buffer that you may have to warm up to 40 degrees for up to 20 minutes if you notice any precipitation prior to use. Here we have some recommended accessories when running the manual RNAscope and BaseScope assays. To the top left are 2 options for washing and handling slides. You may use either the TissueTech wash tray common in most labs, or you can use our Easy Batch Slide processing system, which was designed to streamline your RNAscope and BaseScope assays and save handling time when working with a large amount of samples. The high-VZ hybridization system to the right is designed to maintain the perfect temperature and humidity to ensure optimal and consistent RNAscope and BaseScope staining. Below are two methods for carrying out the heat-mediated target retrieval step required for some types such as FFPE or fixed frozen samples, in which we have the additional protocols through our website. Some general tips to ensure success with the RNAscope and BaseScope assays is to first follow the protocols. We do have many different protocols of which are complete with many details to help address the best way to run the assay successfully. Next, the sample treatment is very important to ensure you get the most optimal RNAscope signal. The pretreatment conditions will differ from sample type to sample type and may require some optimization. Using controls is also highly recommended, especially when you are troubleshooting the assay. And we also note that you can review the checklist that you're using all of the required materials, of which I'll go over on the next slide. On this checklist are some other key components that can help ensure your success with the RNAscope or BaseScope assays. This includes the image hydrophobic barrier pen, ensuring that you have both positive and negative control probes, using the appropriate methods for doing your pretreatment and target retrieval, and ensuring that you have all the appropriate mounting mediums and reagents required to run the assay. For full information, this is all written within the user manuals for the respective RNAscope or BaseScope assays. We also have some general guidelines to follow when you're running the assays. This includes applying all the amplification steps in the right order to ensure that you don't let the slides dry out and that your hydrophobic barrier remains intact for the duration of the assay, and to try not to alter the protocol in any way without discussing with technical support. When running the RNAscope or BaseScope assays, here are a few factors that could affect the outcome of your results. This could be that you have fixation conditions that are not optimal, which may lead to potential RNA degradation. The hybridization conditions may not be as optimal, which include samples drying out during your assay. Or you may even have some special tissues that are sensitive to the pretreatment. To help regard and address these factors or solutions, you can fix the samples as recommended. So we do have recommended guidelines for most tissues noted in our user manual. You could also acquire new samples to help assess RNA quality if you feel like the RNA quality has been affected. Using the high-VZ hybridization system does help to ensure optimal hybridization conditions and to ensure that you're using all of the standard pretreatment and reagents required. This assay is the HD Brown Chromogenic RNAscope assay, which is our most robust assay. It does work well in tissues that may have AP-dependent backgrounds, such as kidney, but we would not recommend it for samples such as lung or skin, which does have some pigmentation. Because it is our most robust assay, it is not sensitive to any alcohol or xylene solutions, and then you can use any xylene-based mounting medium. Here's the HD Red RNAscope assay, as well as the BaseScope Chromogenic assay, which does utilize the same fast red chromogen. Tips include using either EcoMount or VectorMount mounting medium to help best preserve the staining. The chromogen is sensitive to alcohol, and if exposed, may cause some diffusion in your signal. To remedy this, we would suggest that instead of going through an ethanol dehydration, the sample should be dried in a 60-degree oven for 15 to 30 minutes or even more before mounting to ensure that there is no residual water. For the 2.5 HD Duplex Chromogenic assay, we would highly recommend to use VectorMount as the mounting medium. Both of the red and green chromogens are sensitive to alcohol, so they should not be dehydrated prior to mounting. Additionally, the green chromogen can be sensitive to extended exposure to water, which can also diffuse the signal or even cause it to fade completely. Note that for the Duplex assay, we would also recommend that you dry your samples in a 60-degree oven for 15 minutes or more prior to mounting to remove all excess water. As noted earlier, you can split this into a 2-day assay by stopping after target probe hybridization in a 5x SOC overnight solution. Further details of this are located in the 2.5 HD Duplex user manual. Here we have the Fluorescent Multiplex assay. We highly recommend to use prolonged gold anti-fade, as we found that this has best worked to preserve the fluorescence of the RNAscope assay. The fluorophores here are conjugated directly to the label probes, which are provided in 3 different color modules seen in the table here. So that if you would like to use the AMP4 Alt-A mixture, this will allow your C1 probe to be viewed in green with the Alexa 488, your C2 probe would be in orange or the Atto 550, and your C3 would be in far red or the Atto 647. Note that you only requires the use of only one AMP4 alt when running the assay, so depending on your sample, you may only need to choose one AMP4 alt at any given time. Here is the multiplex fluorescent V2 assay, again, which we use the prolonged gold anti-fade mounting medium. This is the TSA-based assay in which you would need to purchase the TSA fluorophores or opal dyes from PerkinElmer. Our recommended starting TSA fluorophore concentration is 1 to 1,500, but we have a working range that you can try from 1 to 750 all the way to 1 to 3,000. This can also be split into a 2-day assay after target probe hybridization in 5x SOC overnight at room temperature. So which assay would be right for you? The first fluorescent multiplex is best used in fresh frozen samples and is made up of the label probes conjugated to the fluorophores. This assay will allow for 3-plexing capability of 3 mRNA markers simultaneously in your tissues or cells. The second assay, the multiplex fluorescent V2, utilizes PerkinElmer's TSA fluorophores or opal dyes and was developed with FFPE or paraffin-embedded tissues in mind to help address the autofluorescence issue. This assay can also be followed up with immunofluorescence, in which we have a technical note up on the support page. With the additional purchase of a 4-plex ancillary kit, the assay will also allow for 4-plex capability to detect 4 mRNA markers simultaneously. The LS multiplex assay is similar to the V2, which also utilizes the TSA Plus or opal dyes, but is completely automated on the Leica Bond RX system. BaseScope is based on the RNAscope technology, but there are several major differences. Here's a general comparison between the 2 platforms. The RNAscope utilizes a probe consisting typically of 20 Z pairs for each RNA target, whereas BaseScope can detect as low as 1 double Z pair or more. The BaseScope probes are designed differently, so they cannot be combined with RNAscope reagents and vice versa. Both RNAscope and BaseScope are capable of single molecule detection, meaning that a single dot represents one single RNA molecule or target. While RNAscope is a robust assay to detect gene expression of most RNA targets, BaseScope is specifically used for the detection of exon junctions, short target sequences, and point mutations. RNAscope is able to detect up to 3 targets simultaneously, whereas currently BaseScope is only available to do 1 target at a time. The assay workflow for BaseScope is similar in that the probes are much shorter in length. It may require a stronger protease pretreatment due to the additional amplification, and there are differences in, again, with the amplification structure, which allows to be able to detect much shorter signal or much shorter probes as opposed to the standard RNAscope probe design. Here is a schematic of the BaseScope assay for FFPE tissues. While it is slightly longer than RNAscope by about 30 minutes, the workflow is very similar and just as easy. The pretreatment is the same as RNAscope with a hydrogen peroxide to block endogenous peroxidases, target retrieval to break up the crosslinks to expose your RNAs, and a protease to penetrate and permeabilize the sample, followed by up-probe hybridization. Your BaseScope preamplifiers and amplifiers are then followed with detection by the same fast red chromogen used in the RNAscope red assays. Here's an example of an application with a BaseScope assay. The experiment takes a look at the complementary determining region in T-cell receptors, particularly the CDR3 alpha and CDR3 beta sequences. There is a very short sequence that is unique to the DURCAT CDR3 alpha and beta sequences in which we designed BaseScope probes. Detection of the CDR3 alpha and beta in the DURCAT T-cell lines are provided in the images below, in which we had 4 BaseScope probes designed to detect both the sense and antisense sequences of CDR3 alpha and beta in the DURCAT cell lines. You'll note that in the DURCAT CD3 alpha and beta probes, we detect expression, whereas in the sense probes, that detected little to no signal. Again, please feel free to contact technical support for questions and any troubleshooting regarding your BaseScope assay. So in understanding the RNAscope assay, we'll take a look at the Semi-Quantitative scoring guideline, which is a scoring guideline to count your dots and choose the scoring criteria based on your dot count with your staining. For more quantitative approaches to data analysis of the RNAscope or BaseScope results, you may visit the link below, in which you can download a free guide describing the different methodologies available to quantify your results. Here, with a semi-quantitative scoring guideline, based on the number of dots that you're seeing in your field of interest, you can then apply a score from 0 to 4. If you have further questions regarding data analysis, again, please feel free to contact technical support and do visit the link below. Looking at the RNAscope and BaseScope assay, you may wonder, what does a dot mean? What is the significance of a dot size? And what is the difference between a dot and a cluster? In this image, with the RNAscope chromogenic assay has been performed with the red chromogen, signal appears as numerous punctate red dots of various sizes and intensities and few clusters. It is the number of dots and or clusters that are important rather than the size of the dot or the intensity of the dot. Each punctate dot equals an mRNA molecule. The small faint punctate dot pointed by the orange arrow is one mRNA molecule. The slightly larger, darker red dot pointed by the teal arrow is also one mRNA molecule. However, it's larger in size due to more double-D probes binding to the target molecule. In contrast, a cluster pointed by the brown arrow is more oblong with a slightly irregular border and results from overlapping signals from multiple mRNA molecules. Similar patterns of dots and clusters are seen in fluorescent versions of the assays, so that the orange arrows point to single mRNA molecules with less DZ probe pairs bound to them compared with the dots pointed to the brown arrow. Now we can take a look at how to optimize your assay. This is how we would recommend to qualify your samples for the assay. As an additional accessory in the event your samples may be limited, we do provide control slides, either HeLa for human or 3T3 for mouse, to run with your control probes to serve as a technique check for your initial assay. If the technique check passes with robust signal and positive control probe and clean background in the negative control, you would then move on to test your control probes in your tissue of interest. This will then serve as an RNA quality check to determine that the RNA has been preserved well prior to testing your target probes. In the event the control probes in your tissue fail, then you may need to optimize your assay conditions. But if you do pass, then we can recommend that you can move on to test your target probes. So why optimize your assay? Here are some potential reasons that you may need to do so. If your samples are underfixed, this can usually lead to over-digestion of your samples using your standard conditions. If you overfix, the signal could then be weak and you may, again, need to optimize your pretreatment conditions. There are also some special sample types that could require different pretreatment conditions and optimizations, with a few listed here. If your tissue of interest has not been listed or you're unsure if the tissue has been fixed per our recommendation, feel free to reach out to technical support and we'll be able to help provide some recommendations. It is always best to start with the standard conditions per the appropriate technical note or user manual for your sample type when qualifying your samples if we do not have a recommendation available. This will help to set a baseline for how the assay performs, and based on your results, we can make recommendations to either scale the pretreatment up or down. Both the target retrieval and protease digestion perform on a sliding scale so that you may adjust up or down depending on what your samples look like or what the fixation conditions are. Optimizing on the automated platforms is also similar in that you would work to change your pretreatment conditions. For the LigaBond RX, this would include changing either your epitope retrieval 2 and protease times, as well as on the Ventana XTR Ultra, which you would then increase or decrease your cell conditioning and your protease. Please, again, feel free to refer to the user manuals for the automation assay workflow and the pretreatment optimization guidelines. Provided that the target is expressed in the sample and the protocol is followed as recommended, the RNAscope assay is guaranteed because tissue samples can be highly variable. Proper sample preparation and the right controls are essential to an RNAscope experiment. Tissue samples must be properly fixed and prepared so that there is a good quality RNA in the sample and no background due to poor fixation. ACD provides guidelines on how tissue should be fixed for optimal performance of the RNAscope or BaseScope assays. Here are serial sections from a human lung cancer sample on which you perform the manual red chromogenic assay. On the left, this is a section stained with our negative control DAP-B, which is a bacterial gene that is not expressed in intact tissue. As observed in the sample, there should be no signal from your DAP-B. In the middle panel is a section from the same tissue sample with our positive control probe for the housekeeping gene PPIB. As observed with this sample, you want to see fairly uniform detection with the positive control probe, which indicates good quality RNA. Certain tissues may express the positive control probe at a higher or lower levels, but in general, you'll want to see uniform expression through the sample. With the results from these two probes, we can have confidence in the test data we see in the following panel. As an example, I'm showing a section from the same lung cancer sample, probed for the immune checkpoint marker program death ligand 1, or PDL1. PDL1 exhibits a wide range of expression in tumor tissues. In this human lung cancer sample, we observed strong punctate dots with the PDL1 probe indicating expression of the PDL1 in the tumor sample. The controls show us that there is good quality RNA throughout the tissue sample and that there is little to no background, so we can be confident that the localized pattern of expression we observe for PDL1 is in fact the correct result. Now we'll go into how to troubleshoot the RNAscope assay. Here in this slide, it shows optimal digestion with a tissue fixed within our recommendations and then treat with our standard conditions. PPIB staining is strong while the DAP-B negative control is clean. Tissue morphology is intact and hematoxylin staining looks to be homogeneous. Note that this particular assay was run on the LigaBond RX, so the pretreatment conditions that are shown are with regards to running this on the automated system. As compared to the last slide, this data set shows tissue that has been over-digested, in which the nuclei are now undefined due to suboptimal tissue morphology, and the PPIB staining is slightly weaker than expected. Again, please note that these conditions were done on the LigaBond RX. Similarly, for underfixed samples, to help remedy over-digestion, you would then try and decrease your target retrieval time. Particularly on the left side, we're looking at samples that are FFPE fixed for 12 hours, so underfixed as compared to our 16 to 32-hour suggestion. Here at the standard 15-minute boil, the Tissue morphology has been destroyed, and you see ghost nuclei, and there's also a high nuclear background, as this is the DAPI negative control that we're looking at, and a weak hematoxylin staining. So in order to optimize this, we've then decreased the boiling time from the standard 15 minutes to the modified 8 minutes, which is then seen on the right side. Here you'll see intact morphology, much stronger hematoxylin staining, and you don't see the background that was seen in the previous sample. Here we're looking at the same tissue section, but also stained with the PPIB. So on the left hand, you'll see the standard 15-minute boil, as opposed to the right, which is the modified 8-minute boil. Again, we're looking at better intact morphology, a stronger hematoxylin staining, and there's even better signal resolution. In the opposite direction, here are some samples that have been under-digested. So the PPIB staining is much weaker than the first two samples, and the tissue morphology looks great, showing no signs of over-digestion. Again, these pretreatment conditions shown here are on the Glycobond RX. Here's another sample of overfixed tissues in which we would need to increase the target retrieval boil time. So on the left-hand side, we'll have a 15-minute boiling time standard stained with PPIB. Again, these tissues were fixed for 72 hours, so way beyond our 16 to 32-hour window. To optimize this, we've then increased the boiling from 15 minutes to 30 minutes, in which you can now see much better signal intensity and signal resolution. When looking at tissue fixation, again, to the left is our optimal condition for fixation with 24 hours, compared to the right panel, which is FFPE tissue that had been fixed for 3 weeks. So as you can compare with the staining, the signal is much lower in the overfixed sample. And so to optimize this, we would suggest to try and fix these samples within our recommendations. Here's an example of what you should see with successful staining of the fluorescent multiplex assay using the 3-plex positive control probe. Here we have the human polar 2A in green with the Alexa 488. In the red is the PPIB with the Atto 550, and then in white is the human UBC marker in the Atto 647. So this was done on the HeLa control cell pellet with the 3-plex positive control probe specifically for human, and also this was using the AMP4 Alt-A configuration. To look at some troubleshooting, well, here's an over-digestion within the fluorescent assay. Majority of the customers for the fluorescent multiplex assay may use fresh frozen samples, which does show how protease pretreatment can affect RNAscope signal quality. On the right, this is mouse kidney sample where you can see there's quite a bit of tissue autofluorescence. But when samples properly digested using protease 4 at room temp for 30 minutes, you can see distinct green and red dots with the control probes. On the left, protease digestion is performed at 40 C, so the tissue is over-digested, and not only you don't have any punctate signal, but you also see an increase in the background. On the bottom is the fresh frozen brain sample. With properly treated samples, there is no background and beautiful punctate signals. And again, with tissue over-digestion, we see an increase in background and a slight decrease in signal. One known issue with the fluorescent assay is that due to the long fixation time, FFPE samples are prone to inherent autofluorescence, which poses a challenge for image analysis. If you use FFPE, if you have background correction software, this can help to reduce the autofluorescence. But alternatively, we now have developed the multiplex fluorescent V2 assay, which helps to address this issue. If you are still experiencing autofluorescence issues with your assay, please do contact technical support. Here are a couple of troubleshooting issues with regards to imaging with the fluorescent assay. This could mean you using the incorrect filter setting, or perhaps doing a longer exposure in which you can use the correct filter settings based on the recommendations within our user manual. If possible, if you have an imaging enhancing software, this could also help to reduce some of the autofluorescence and high background. If you think that it's possibly due to compromised RNA quality or sample preparation, you can try and prepare your samples per our recommendations in our user manual. We would always suggest to perform your assay with the positive and negative controls, as this helps to best assess RNA quality. Another common troubleshooting would be for sample detachment. So in particular, we're looking at tissue detachment with a standard protocol. Potential cause could be that the wrong slides were used, in which we would highly recommend to use Superfrost Plus slides. This could also happen if it's due to suboptimal tissue preparation. So again, we would always recommend you prepare your samples based on our ACD recommended guidelines. You could even bake slides for a longer time, depending on which part of the assay, of which I'll go into further detail for some sample preps here. Here we have the sample detachment for the FFPE samples, in which the modifications you can make would be to do an additional baking step after your deparafffinization. You can also do an additional baking step after your target retrieval. Here with fixed frozen samples, we have 3 different modifications in which you could dry your slides longer at -20 after cryosectioning. It says in our user manual for 20 minutes, but the longer you dry your slides, the better. So we can suggest to increase up to 2 hours. After doing cryosectioning, you can also post-fix your samples in 4% TFA for additional time. And you can also do a baking step after the target retrieval. In the event that the first 3 modifications don't work, you could omit your target retrieval and extend your protease time. But before you do so, please feel free to contact and consult with technical support. Quickly, we'll go over the dual RNAscope ISH and IHC compatibility. One of the most asked questions is, can you combine the RNAscope ISH with IHC or IF? And the answer is yes. Both procedures do have a similar workflow that requires sample fixation, some kind of pretreatment to unmask your RNA or protein, and subsequent detection. The recommended workflow is that you would pretreat and permeabilize your tissue or cells, and then depending on the tissue, it may require both a heat-mediated target retrieval as well as a protease digestion. You would then complete the RNAscope ISH portion and then follow with the IHC staining after. We do have some recommendations to ensure success with your dual ISH, IHC, or IF assay. First of all, the dual ISH, IHC protocols require optimization. In general, it is recommended to combine a working RNAscope protocol with a working IHC protocol. In addition, you would work with antibodies and a protocol that are known and already established with your tissue samples. Second, it is advisable to perform ISH first followed by IHC. Third, it's advisable to optimize the IHC assay separately using the RNAscope pretreatment reagents to ensure your protein can still be detected following the RNAscope pretreatment. RNAscope does require the protease digestion to unmask the RNA, so do keep in mind as you optimize your IHC. Lastly, the dual ISH IHC assay works better for highly expressed proteins due to the protease treatment that is used during the ISH protocol. We do have technical notes available on the support page to combine the ISH with IHC or IF protocols. And if you have further questions regarding ISH IHC, please do contact technical support. Here is one example of using the RNAscope singleplex brown assay to detect the OLSM4 probe with subsequent IHC staining in red for lysozyme. Another example of the dual RNAscope assay with 2 mRNA markers followed by immunofluorescence staining. The IDO1 and PDL1 RNAscope markers are stained in green and red, respectively, while the CD8 antibody is stained in white with Cy5 using the multiplex fluorescent V2 assay. Briefly, I'd like to provide a quick introduction to our latest released RNAscope assays on the Ventana Discovery platform. The VS Universal Assays for Roche's Discovery Ultra are 3 separate products leveraging Roche's Universal Protocol. The VS Universal HRP is a single-plex brown assay, allowing the ability to run ISH and IHC in a single run. It also enables single-plex fluorescent ISH and immunofluorescence. The VS Universal AP assay is a red assay, allowing ISH and IHC in a single run. And together, they also can conform to put the Duplex assay, which is a red-brown combination to identify two targets simultaneously. The table on the right shows a high-level comparison of the new VS Universal assays as compared to our current 2.5 VS red-brown assay. A notable difference is the ability to then combine with the IHC in one single protocol. Here we are showing universal HRP staining with lymphocyte activation 3, gene 3, a cell surface molecule, AP staining with a PD-CD1 and immune checkpoint marker, and then both targets together in the VS Duplex assay. To demonstrate the contrast and robustness of the HRP and AP, we show staining in lung cancer tissues that have black deposits from the lung tumor carbon deposits. And here we're showing keratin-19 using VS HRP. Keratin-19 is a marker for tumor cells in the lymph, blood, and bone marrow in breast cancer patients. Using the VS AP, we've stained with endothelial cells with PCAM1. And the last image is the Duplex combination. This assay also allows for the capability to stain a single-plex fluorescent assay using the VS Universal HRP kit and Roche's fluorophores. This allows for the capability to combine the single-plex fluorescent ISH with immunofluorescence with your protein of interest in a single protocol. Alternatively, we can also do this in a chromogenic ISH-IHC. So this is using Ventana's red and yellow chromogens to be able to do ISH with PPIB and IHC. So I'd like to address a couple of frequently asked questions. The first being, do I need to run every single AMP reagent when doing the multiplex fluorescent assay? And the answer is yes. Each AMP reagent serves as a building block for the amplification structure of your target mRNA. If any AMPs are missed, this may result in no signal. So if we look at the amplification diagram to the right, each of these amplification reagents within the kit do perform some sort of tree or a branch to the amplification structure, so that if you are missing any of the AMP reagents, then potentially one of these portions cannot be bound, and therefore you will get no signal. Another question is, what does the C1, C2, and C3 nomenclature mean? So previously I had addressed this, that in order to detect multiple mRNA simultaneously, multiple amplification trees must be built. With that in mind, how do we ensure specificity? So to the double Z probe design, we have a tail sequence that is indicative of a channel designator. A C1 probe has a specific C1 tail sequence that will only allow for binding of the subsequent C1 pre-amplifier, amplifier, and label probe molecules. The same can be done for the C2, C3, and even C4 in a multiplex assay. This image shows the distinct yet separate amplification trees for each channel in the fluorescent multiplex assay, which allows for simultaneous detection of multiple markers. To go over a couple more frequently asked questions, are RNAscope assays compatible with different tissues and sample types? And the answer is yes. And the assays can be used with FFPE, fresh frozen, fixed frozen, and even cultured cell samples. The RNAscope automated assays do primarily support FFPE tissues, so it can be compatible. We would still recommend that you contact support for how to optimize. Another question is, why would I not see signal with my positive control PPIB? Going through our troubleshooting and optimizing, it could be due to compromised RNA quality because the RNA has likely been degraded. It could also be that your sample preparation and pretreatment have not been optimal. In this case, you may need to optimize your pretreatment or even perhaps re-prepare samples to ensure RNA quality is good. Another question is, how flexible is it to switch fluorescent dyes between channels in the LS multiplex fluorescent assay or in the V2 multiplex fluorescent assay? And the answer is that it's actually quite easy and flexible to do so, as the fluorescent dyes are done in separate steps in the multiplex V2 assay or the LS multiplex. And so your C1 does not have to be tethered to the fluorescein, C2 does not have to be tethered to Cy3, and that since they are independent steps, you can swap accordingly per your preference. Again, another commonly question is, can you combine the RNAscope and BaseScope assays so that you can look at your shorter targets or splice variants along with your standard RNAscope? And the answer is, unfortunately, you cannot do so, as the chemistry between the two is completely different and not compatible, from the probe design even through the amplification structure. For additional information, please visit our website at acdbio.com. If you click on the Support tab, there are multiple pages that have a wealth of information, including troubleshooting help, user manuals, tech notes, training videos for the manual assay, as well as previously recorded webinars which cover a wide range of different topics and applications. With that, I'd like to thank you for attending today's webinar.